Hi everyone, welcome to my overview of the article Antidepressant Drugs Act by Directly Binding to TRKB Neurotrophin Receptors. The article we're going to be looking at today looks into the effects of antidepressants and how they exert those mechanisms and those impacts. So just a quick overview of depression. 6.7% of adults in the US either are currently suffering or have suffered from depression, and so that's a huge number. So obviously this is an important issue. It was found that depression does have a genetic basis, and symptoms of depression can include mood changes, sleep changes, fatigue, thoughts of suicide, and many more horrible symptoms. In terms of its physical changes on the brain, these are mainly concentrated within the limbic system, and so you see a reduction in gray matter volume of the hippocampus as well as the prefrontal cortex, and you also see an increase in the amygdala size. So the current treatments for depression are not well understood. We have SSRIs and ketamine. SSRIs take a long time to work, whereas ketamine doesn't, and that is not very well understood because they both have a similar mechanism of action. We know that they work best alongside therapy. We also don't completely understand why. People have looked into the binding of BDNF and TRKB and how that may be involved, but it's still not well understood. So in this article, overall what they discovered was the effect of cholesterol in the process, as well as having a more definitive proof of what the effect of antidepressants are on TRKB and BDNF. Which is the brain-derived growth factor. And so here are the main things that they looked at. We're going to look at this first one in much more detail in the upcoming slide, but as for the others, they basically looked into the antidepressants binding to the transmembrane domain, and so they found proof that the antidepressants were actually binding to the TRKB receptor. They also found that antidepressants promote the membrane trafficking of TRKB, and that this also led to an increase in AMPA receptors on the cell surface. And so this kind of has a positive cycle of then increasing TRKB receptors available to bind to more BDNF. And they also found that binding to TRKB mediated the antidepressant-induced plasticity that we see in brains. And then we will also look into this last detail here in the upcoming. So for cholesterol, their main experiments found that an increase in cholesterol led to an increase in levels of phosphorylation of TRKB by BDNF. So basically cholesterol enhanced the effect or even allowed for the effect of BDNF on TRKB. The first experiments they did was through computer simulations of rebuilding the TRKB receptor and finding areas where it could directly bind with cholesterol. And what they found, which you can see here in figures E and A, is that there was a direct binding motif on TRKB for cholesterol. And so in figure E, we can see that as cholesterol increases, the wild-type TRKB receptor has increased binding, whereas the mutated motif location does not see an increase in binding. So this is incredibly important. Cholesterol treatment increased cell surface translocation of TRKB. So this was another important aspect, which is that cholesterol led to the effect of BDNF that we saw with increasing its translocation to the cell surface. The BDNF effects in general, though, of TRKB on TRKB were inhibited by pravastatin, which is a cholesterol synthesis inhibitor, and they were rescued by cholesterol but not by BDNF. So this is incredibly important because it kind of implies that the effects that BDNF has on TRKB are kind of allowed for through cholesterol, and so cholesterol is a very important aspect in those effects. Finally, we found that mutating the cholesterol binding motif region on TRKB did not reduce its binding affinity to BDNF, but it did still reduce the TRKB phosphorylation by DNF. And so this also tells us that cholesterol is critical in BDNF's ability to phosphorylate TRKB and kind of signal that cascade, which is what you generally see with the TRKB effect on depression. And also its translocation to lipid rafts, as we said earlier. The behavioral effects that they were able to link between antidepressants and BDNF and TRKB were super important. So they conducted several experiments to do this. The first one is that they put mice in a training arena where they had specific visual cues as well as objects, and after a specific training period, they had the mice, they moved one of the objects and they tracked how long the mice spent in the old object's location versus the new object location. So what they found here, as seen in figure A, is that mice with a wild-type TRKB receptor were able to perform well when they were given the fluoxetine treatment, whereas mice with the mutated cholesterol binding site of the TRKB receptor, which, as we saw from the last slide, sort of left the TRKB receptor kind of useless, did not see the improved ability in the task. Similarly, this lack of improvement in the task was seen in BDNF haploinsufficient mice and mice treated with pravastatin, which is the cholesterol synthesis inhibitor. And so what this proves is that BDNF and cholesterol are both critical in having that behavioral effect. Interestingly, serotonin transporter knockout mice, so what was originally thought was the main effect of SSRIs, they responded normally to the fluoxetine treatment. So they responded the same as wild-type mice here. So that kind of implies that the serotonin transporter aspect of the SSRIs may not be important at all, at least in terms of this behavioral aspect. In a kind of similar test, they put mice in a cylinder of water for 6 minutes and they measured their immobility. So this is kind of their fear as well as their sort of anxiety response. And so in this test, they found that mice treated with fluoxetine and ketamine, the wild-type mice, were able to have reduced immobility during the test, but mice with the cholesterol motif having been mutated did not find that reduced immobility, and so they were more prone to having those fear and anxiety responses during the test, and that is what you see in these two figures here. And finally, they had a conditioned fear response where the mice were given shocks in a specific context, and then they were conditioned to that, and later they were brought into the same context without the shocks, and they measured how long the mice kind of froze up in that fear-based position. So what they found here is that after treatment with ketamine and fluoxetine, the wild-type conditioned mice did not had a reduced freezing response, and so that's what you see over here and over here. And interestingly, and of course, the TRKB cholesterol motif mutated mice did not have an improved freezing response, so they saw an increase in their fear response, even when they were given the fluoxetine and the ketamine. And the mutated mice and the wild-type mice who were treated with pravastatin didn't see a response at all. So both of them didn't see a response when they were given the fluoxetine and the ketamine, and that's what you see over here. And so only the wild-type has the improved response to fluoxetine, whereas both the wild-type and the mutated TRKB receptors didn't have an improved response when they were also given that cholesterol inhibitor. So again, just kind of reiterating that cholesterol is crucial in these effects of BDNF and TRKB. In conclusion, the article has demonstrated that antidepressants likely mainly, if not very importantly, act through their effects through the effects of BDNF on TRKB receptors, and then also that cholesterol has a crucial role within that. This knowledge can have very large implications in terms of future drug development, as well as a greater push for therapy with more science-based reasons. Thank you so much for listening, and just finally credit to the authors of the experiment. Here are their names. Thank you so much.